Hello, everyone. Welcome to today's live event related to nitrosamine analysis. I'm Brian Demansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll serve as your host in the discussion about the state of regulations, how and when to consider the four procedures of USP General Chapter 1469, and examine the challenges associated with the wide variety of drug substances that may require analysis. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the related content list with clickable links relevant to the material being presented today, including links to our nitrosamines website and example applications for each of the four procedures. On the top left of the slides is the question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the question box are survey questions that you may fill out anytime during or after the presentation. If you would like to be contacted by a Shimadzu representative directly, use the Request Demo section to start that process. The icons at the bottom of the screen can be used to restore any of the widgets in case they are minimized or hidden. All right, let's get started with today's presentation, so we can find out what it means to be nitrosamine ready. In order for us to find good analytical solutions, we first need to define the problem. So we need to look at what is a nitrosamine. A nitrosamine is a nitroso-functional group that has been modified to any primary, secondary, or tertiary amine. And the FDA and ICH recognize compounds that contain these moieties as potentially mutagenic. When we consider developing analyses for these types of compounds, there are several challenges that arise. First and foremost is that the impurities in question may be in much lower abundance than the API or other related impurities. There can be a number of formulation elements, including excipients, that may present challenges for separation and therefore interfere with sensitive analysis. And that need for high sensitivity is certainly present given the acceptable daily intake limits that have been set by regulatory agencies. While pharmaceutical interest in nitrosamines is relatively recent, concern over these substances is nothing new. In the 1960s, at the dawn of the EPA, over 300 nitrosamine substances were investigated in rodent cancer bioassays, with over 90% of them being deemed carcinogenic. There's currently at least two methods, including method 521, that are used to examine environmental samples, especially groundwater and surface water, for nitrosamine presence. Current concern regarding nitrosamines in pharmaceuticals began in 2018, prior to the SARS-CoV-2 pandemic, and was first noticed in the EU with nitrosamines appearing in sartan drugs. That year, the FDA announced that there would be voluntary recalls on valsartan because of the detection of NDMA. Later that year, the FDA posted GCMS single quad test results showing NDMA and NDEA levels in the recalled valsartan products. By September of the next year, nitrosamines were also being detected in other over-the-counter medications like ranitidine. This led to further regulation of NDMA in metformin in 2019, and the number of drug substances continued to grow that were of concern. With the number of drug substances growing, the need for clarity in regulation became clear, and over the next 2 years, there was a shifting of responsibilities to the regulatory agencies, with the first step being the FDA releasing LCMS-MS and LC high-res mass spec methods as a draft guidance. There was then an agreed-upon alignment between USP and FDA to begin the development of a new general chapter, Chapter 1469. As the number of drug substances that were identified continued to grow in 2020 and 2021, the general chapter was completed and went into effect on the 1st of December 2021. While the implementation of General Chapter 1469 provided methodologies and potential targets for small molecule nitrosamine impurities, there was one big problem still remaining, and that was how to address API-related nitroso compounds. In order to do this, the FDA introduced a guidance in August of 2023 regarding NDSRI, or nitrosamine drug substance-related impurities, and their acceptable daily intake limits. This has resulted in the need for analysis and risk assessment to be planned and implemented by the 1st of August 2025. Based on the FDA guidance, one of the most important steps in developing your nitrosamine analysis strategy is performing the risk assessment on each of your drug substances and products of importance. In order to do this, the FDA guidance provides a flowchart to determine the potency category of any substances that are part of your drug product. This allows an intake limit per day to be established. This does require ongoing assessment anytime that changes are made either to synthetic route, formulation, or to packaging interactions. Based on the determined potency category, the acceptable intake limits range from as little as 26.5 nanograms per day up to 1,500 nanograms per day, thereby making a large dependence on the need for sensitive analysis surrounding the amount of drug substance that will be consumed each day. Because of this relationship between the daily acceptable intake level and the maximum daily dosage, it's important to consider how much of a drug substance will be prescribed to a patient when trying to determine the LOQ that's desired for that method. We can determine a basic upper limit by doing the simple calculation of acceptable intake divided by maximum daily dosage. This will give us a maximum target, and if we look to be one order of magnitude below this for our LOQ, then we can choose an acceptable level. In this case, if we consider a drug that is dosed at a maximum daily dosage of 880 milligrams per day, when we're considering NDEA, we need to have a detection availability of no less than 30 ppb, and we would like to have an order of magnitude better. So if our assay can perform at 3 ppb for an LOQ of NDEA, this would be acceptable for a drug dosed at 880 milligrams per day. When we look at how that translates for potency category 1 and 2 substances, we can see that there is a great dependence on that maximum daily dosage in terms of our goals for our limit of quantification. These detection limits are much easier to attain for drugs that are dosed in small amount as a maximum daily dosage limit. You can see here the difference in those target limits for a 40 milligram per day drug substance versus 880 milligrams per day. And it is because of this wide range based on that maximum daily dosage rate that we do require sensitive analysis. Remember, as we look at these limits in ppb by maximum daily dosage, that is to reach the maximum acceptable intake. In real terms, we want to see limits at our instrument from our tested substances that are at least one order of magnitude below this to ensure safety of our drug products. Now that we understand some of the background, let's look more closely at the 4 procedures outlined in USP General Chapter 1469. While there are 2 LCMS and 2 GCMS methods, in this discussion we'll concentrate primarily on the challenges associated with the LCMS methods and mention in brief how and when to use the GCMS methodologies. Procedure 1 details LC high-res mass spec methods and includes targets like NMBA, which was one of the first substances identified as being unsuitable for GCMS analysis. So in cases where NMBA is a substance of concern, LCMF methodologies, Procedure 1 or Procedure 3, would be recommended. Chromatographic guidance is provided in terms of mobile phase, stationary phase, and a method that was developed for a particular drug substance is provided as example in USP General Chapter 1469, but those chromatographic requirements are not binding, as we'll see later. Why would you choose LC high-res mass spec? Ultimately, it's for the accurate mass ID of nitrosamine impurities and discovery of NDSRIs. It may not be the best tool for routine analysis, but it's certainly important in the investigation of developing your nitrosamine analysis profile. So let's look at some data obtained using a Procedure 1 based method. Here, the mobile phase and stationary phase are USP L1 and water and methanol with formic acid as the mobile phase system, as well as general instrument parameters. When we look at these chromatographic results using just standards, we can see that there may already be some things that will challenge our analysis when we introduce more complex formulations down the line. In this case, we can see by adding a couple of extra compounds like NDPA, we've induced coelution between NDPA and NDMA. This could be an interference that may cause inefficient ionization of the source, or it may increase our limit of detection or limit of quantification to a level that becomes unacceptable. One other thing to note is that NDIPA and NDPA ultimately have the same daughter ion that's being used for analysis. This can also present a complication if we don't have significant enough chromatographic resolution. Despite these challenges, though, the calibration curves still indicate that with this separation, we can achieve low enough limits to satisfy our need for sensitivity. If instead, though, we choose to use a different stationary phase or potentially a slightly different mobile phase system, we can induce very different separations. In this case, we're showing representative data from a USP L43 column, and we've traded off. Instead of having certain compounds coelute, we've now chosen to obtain greater sensitivity for NDBA at the sacrifice of now having NDPA and NMPA coelute. There is, though, this type of flexibility built into the general chapter, so that whatever your nitrosamine profile is for your compounds, you can find an acceptable separation and analysis method. As we look at these summary results for a Procedure 1 method, take note that for each of the compounds, the LOQ is no greater than 1 ppb for all of these substances. As we think back to the chart we saw relating acceptable intake to maximum daily dosage, this may seem like a much lower LOQ than is required. However, that table represented the maximum amount that would be allowable for exposure. Certainly, drug manufacturers always wish to be below that, and if they can prove that they are below that by more than an order of magnitude, the incumbency for ongoing testing requirements may be mitigated. Here again is a brief summary of LOD calibration range, indicating that LOD and LOQ are again well below the AI limits prescribed. Because of the relative size of nitrosamine impurities, there are many small molecules whose isotopic variants may also present mass interferences. Owing to its common use in synthetic processes involving amines, DMF is of particular concern as it has isotopic variants that may present both coelution problems as well as mass identification problems for other nitrosamines. Certainly, the direct way to solve this problem of NDMA and DMF is to achieve on-column resolution, which, using the method illustrated in the slide previous, we were able to achieve. However, if we were not able to get that on-column resolution, then overestimation of NDMA becomes quite possible if we have DMF as part of our substance. You can see here, though, the extracted chromatograms showing the relative abundance of the different isotopic variants of DMF, including the non-detect of the 15N form. Looking at the results from actual formulations, we can see that DMF is indeed detected in the drug product and at a significant amount. So if we weren't able to accomplish that on-column resolution, there may be significant overestimation of NDMA. So the importance of Procedure 1 was to help us determine a profile for initrosamine analysis, including identification of unknown compounds or new undiscovered NDSRIs. Once we know what our profile is, though, we need an even more sensitive tool, potentially, to do targeted routine analysis. Procedure 3 gives us that tool in the body of LCMS triple quad, and by using a similar analyte list to Procedure 1, but adding the augmentation of having deuterated internal standards in order to enhance that quantification that we're seeking. So let's take a look at an example method that we could conceive of for LCMS triple quad. In this case, we're using APCI ionization and using a biphenyl stationary phase along with a water, methanol, and formic acid mobile phase system. By using that biphenyl stationary phase, we can see that we end up with quite a bit longer method that has significant retention for most compounds, with the exception of NDMA. This provides much better on-column resolution of many of the species, and if we are trying to develop an assay that will allow for a wide range of drug products, a chromatographic approach like this may be quite useful, as there's plenty of space for unknowns or other nitrosamine-related impurities to elute in that space and avoid coelution issues. As with any chromatographic method, the performance is only as good as its repeatability. Here we see results for system suitability testing, and we can see that for each of the compounds of interest, the FDA guidance requirement of RSD being no more than 10% is more than easily met for all of the compounds. This assessment was performed at a 3 ppb standard concentration. And while those results are easy to achieve with neat standards in solution, the real challenge comes in analyzing drug substance and drug product. When we do that, we find that we are still able to meet those LOQ requirements for the regulatory levels. However, when we look at real samples, there does become a greater variability in that spike recovery. Particularly the variance in NDMA does suggest that there may be another issue at play. So as we looked closer at these results and started wondering why is there this variance in the NDMA, we realized that there was a problem. And if we look at these chromatograms that include both a blank as well as several calibration levels, we can instantly hone in on that problem. Even the blanks for NDMA do not have 0 content. So we were wondering, where is this interference that's appearing as NDMA coming from? And through some difficult analysis, it was determined that the nitrogen generator was discovered to be the source of the contaminants, and that the contaminant itself is not NDMA, but possibly an isotope of N-butylamine or diethylamine. Since it would be impractical to choose not to use nitrogen generators, of using an inline hydrocarbon trap that integrates directly to the instrument. With the hydrocarbon trap in place, we can clearly see that without the filter, our baseline noise count is far too high to see the actual data coming from the NDMA peaks. Once the hydrocarbon trap filter is installed, baseline noise is reduced from about 60,000 counts per second to only 2,500 counts per second, allowing easy detection of the transition of NDMA. But enhancing the purity of the nitrogen, thing that was looked at. In terms of sharpening the peak, giving it increased height versus width, the initial B concentration decreased from 5% to 1, resulting in a 2 times baseline reduction. The voltage biases within were properly adjusted as well to increase intensity of the signal and as well to reduce baseline. By changing as well on the unit from high to low, there was an additional improvement in the intensity of the signal. When we apply this holistic approach to method optimization, we can see a great improvement in the LOQ for every one of our instruments in our lineup. By making those method changes, by taking the physical mitigation efforts with regard to the nitrogen supply, we've actually been able to achieve with our LCMS 8045 a 0.27 nanogram per mil LOQ, with the 8050 0.22 nanograms per mil, and with the 8060 NX 0.1 nanograms per mil as our LOQ. So as we conclude our discussion of procedures 1 and 3, there are some things that are common to both that are considerations of the LC portion of the system that should be made. First and foremost among those is that difference in the relative abundance of the API versus its impurities. Oftentimes that means that we may need to do a different injection scheme, or we may need the ability to divert that large amount of API prior to it entering the mass spec. So in some cases, to detect low-level impurities, we may need larger scale injections on the order of 20 microliters or even up to 50 microliters as an injection. And the ability then to see very large amounts of substance at the same time that we see small amounts, or to perform again 2 injections and divert large amounts of API to waste so that they don't saturate our detector. That divert valve can be an important piece because we also may need to get rid of highly polar salts or sugars if we're dealing with serum samples. And we don't want, again, to saturate that. And in terms of determining valve timing, a UV detector may be a useful addition if we are doing that dedicated impurity analysis. Our API will be visible at concentrations appropriate for a UV detector, while at the same time we're able to attain the sensitivity we need for small level impurities. While the majority of our discussion today has centered on the challenges of the LCMS-based methods, procedures 2 and 4 define GCMS triple quad methods that do provide an alternative path. 2 is Headspace GCMS-MS and allows for rapid introduction of samples, minimal preparation, and the benefits that come along with GCMS. Procedure 4 is intended for injection of crushed tablets or granule material and does not require any special application of the samples. However, because of using a direct injection methodology, more interferences may be present. It is required then that a more sensitive GCMS triple quad mass spec be used for that procedure 4. So why are there 4 different procedures? Why is there the option to do LCMS versus GCMS? Ultimately, the decision comes down to what's appropriate for your analysis profile, and it's picking the right tool for the job. So instrument cost can be lower. We can often experience higher intraday repeatability, reduced complexity, no mobile phase prep, simplified sample prep, and reduced interference from non-volatile formulations. With LCMS, if it's dissolved, it has the potential to interfere. With GCMS methodologies, if it cannot be volatile, it will not travel through the GC column. Obviously, though, we do have to have some trade-offs. So not all NDSRIs are appropriate for GCMS analysis. Any compounds that are labile or may convert between species with the addition of heat are really going to be appropriate for GCMS. Also, GCMS LOQs, as demonstrated in our example methods, the provided guidances, are higher than the LOQs provided for by LCMS. So for the most sensitive analysis products, LCMS may be the more viable consideration. However, for drug substances and compounds that do not have such low limits of detection or quantification, GCMS is a great tool. What we need to do is look at this problem. And making that right choice does require understanding of each drug product and the associated nitrosamine risks thereof. So we've discussed at length the procedures that are available for analyzing nitrosamines, but let's take a step back for a moment and consider where are these nitrosamines coming from? And certainly there are a number of different potential sources, including the development process and the synthetic route chosen for a drug. Based on that process, there's an increased or decreased likelihood of the formation of nitroso compounds. Manufacturing processes, as we go from that laboratory scale synthesis to process development, changes or compromises may be made in that synthetic route that result in additional exposure to risk of formation of these compounds. The particular solvents used in those processes may themselves contain impurities or may contain precursors that when they interact with other substances or formulary elements can lead to nitrosamine formation. Excipients, other components of the drug formulary, may either be sources directly themselves or provide a seed for interaction. Nitrates in water used for synthesis or for processing, or that may be residual on packaging materials, can potentially present an issue for interaction and transformation after the production of a drug product. Storage conditions, environmental concerns, exposure to moisture and heat, as well as the combination of all of these concerns, is certainly a potential source. So that is to say that while we've addressed the idea of doing a risk study, setting up analysis profiles, and carrying out sensitive analysis of nitrosamines, all of these factors and their shift may change that nitrosamine profile. So be aware that nitrosamine testing, while we're entering into an era of it, is going to be evolving. And we should all be anticipatory of regulatory changes that will continue to happen over the next 3 to 5 years. And as we look at source mitigation concerns, we can find other targets for potential analysis. We may need to be doing routine ion chromatography analysis of precursor ingredients to detect nitrates and nitrites before they enter the drug production supply chain and therefore become a potential source of nitrosamines in finished drug products or drug substances. So understand that while we have some guidelines, the world of nitrosamines is going to continue to grow and expand, and the ways in which we address the problems beyond just detecting them may evolve as well. Quiet now. As we wrap up today, I want you to take away some of these key ideas: that designing and implementing LCMS methods and GCMS methods for nitrosamine analysis at trace levels faces several challenges. First off, predicting and identifying NDSRIs, avoiding formulation interferences, isotopic interferences, manufacturing solvents, and gas purity. Patient daily dosage has a meaningful impact on sensitivity goals. And understanding the nitrosamine profile of each manufactured drug is key to choosing the correct analytical tools. While we are experiencing a period of certainty in terms of the need to assess these risks and report a strategy to mitigate their harm, this is only good until August 1st of 2025. And as this topic evolves, we expect that there will be further regulatory action over the coming years. So the takeaway there is stay tuned. Nitrosamine analysis is here. It's not going away. And now we have clear deadlines to implement strategies that need to be addressed. And we hope that Shimadzu has presented you 4 compelling solutions to help you solve these analytical challenges. As we transition to our question and discussion period, I certainly encourage you to enter in any last-minute questions you have to the question widget in the console, and certainly encourage you to seek additional information either at our website, solutionsforpharma.com, or by using the links below to access the Nitrosamines website or any of the 4 procedure application notes. I thank you, and we will proceed now to our question and answer session. First off, let me say thanks to everybody for participating today, and again remind you that if you have any questions at this moment, go ahead and put them into the question widget in the console, and let's jump right into the questions that we do have. One of the first ones that has come in was in regard to GCMS methodologies. There was a question about the meaning of direct injection GCMS-MS method with regard to procedure 4, I'm presuming. And really, we are using the term direct injection not to mean the same thing you might say with a pyrolyzer or with a method where my sample introduction is directly to the source of the mass spec or to an adaptation onto the source, but it's meant simply to distinguish from the headspace methodology of procedure 2. So this would be a traditional way of preparing a sample where I would take a tablet or a solid formulary and use a mortar and pestle to comminute and then dissolve into an appropriate solvent for GCMS injection. And because you are then taking that whole tablet rather than doing the headspace sampling of the matrix itself, you may potentially introduce more interferences. So when we talk about the appropriate type of GCMS-MS for procedure 4 to go with that direct injection strategy, that would be a more sensitive model of GCMS-MS that would be required. All right, next question I'm seeing, let me phrase it this way. It's asking if there are labelability concerns with nitrosamine compounds, why are there GCMS options available rather than having LCMS options be the sole and best option? That really does come down to the risk that each individual drug substance and drug compound presents. And by doing an appropriate risk assessment, you can determine whether or not the compounds that need to be subject to ongoing testing are going to be appropriate to assay by GCMS-MS methods, procedure 2 and 4, or if they would require, say, procedure 3, that LCMS triple quad method that would provide the best sensitivity and allow for the widest range of molecules, including those that are considered labile. But as was addressed kind of in the pros and cons of LCMS versus GCMS, certainly the level of operator experience can be a little bit lower for a GCMS triple quad method because there's less that goes into mobile phase preparation. The separation portion tends to be a little bit more reliable rather than having the added complexity of liquid chromatography. All right, we have some other questions that are coming in still. So there was a question that came in regarding the use of ultra-high purity nitrogen tanks as an alternative for the baseline issue. And in fact, that's a wonderful suggestion. That is what was used in order to discover that the source of the observed impurities was from the nitrogen generators. We see that, though, for laboratories engaging in ongoing testing as not necessarily the best solution, especially if the LCMS is used with high gas flows in order to achieve high sensitivity or used for extended periods of time. Now, if this is something that's being done as an infrequent assay, that is most certainly an acceptable alternative. Certainly, the combination of the two would not be undesirable either. Adding on the inline filter simply adds capacity in case your gas supplier may not have supplied the proper purity, and it can just give you additional prophylaxis against being out of specification with your designed assay. Okay, the next question that we're seeing is related to LCMS assays, and it's regarding one of the example chromatograms that was shown where APCI ionization was utilized. ESI ionization can be used, and choosing which ionization method is appropriate for your compounds and provides the required response versus sensitivity is part of the method development process for LCMS. That's another aspect of the increased complexity that comes with LCMS, but again, then the increased flexibility. So you do have no constraint within methods themselves. It's really a target to meet in terms of sensitivity goals. All right, let's see. We have a couple other questions here. So the other question was relating to USP involvement. And it asks, will there be USP monographs for specific nitrosamine impurities in specific drugs? The answer to that is both yes and no. So prior to the current crisis surrounding nitrosamines, there were already some identified drug substances where the USP monograph for those non-patent protected drugs included specific observation of nitrosamine compounds, mainly due to their known presence in the synthetic route of those drugs. The recent problems that have been seen and the adoption of General Chapter 1469 hints that there will not necessarily be direct monograph guidance for these specific impurities, which is why that dovetails as well with the idea that the FDA has released the MDSRI guidances, indicating that the identification of potential nitroso compounds related to the drug substance is part of the drug filing process. So it's uncertain in terms of will there eventually be monographs for many of these substances that contain their identified nitrosamine impurities? Maybe. Time will tell, I think, on that. But the approach that's being adopted by the FDA and USP allows for the reporting of those substances and the accounting for them without having to have a dedicated monograph that prescribes very specific chromatography. I think as well the sensitivity requirements and the abundance of these nitroso compounds has dictated that they require mass spec methodologies. It would certainly be a major sea change for USP to start including mass spec analysis as part of monograph requirements. Okay. I think at this point we have unfortunately reached the end of our allotted time for today. So I would encourage you, if you still have additional questions, to go ahead and put those into the widget. If we don't have time to address them here in the live session, we will reach out to you each individually. And once again, I want to thank everyone for attending and participating. There will be an email link that was sent to the email you used for registration to allow you to review the recorded version of this presentation at any time. Thanks again for your attendance, and we encourage you to attend our upcoming webinars. check solutionsforpharma.com for the latest updates, and have a wonderful day.